So now I'd like to introduce you to some of the basic shapes that we'll find in bacteria. Again, this is a foundation slide. Pay close attention as we go through. Pay close attention to not only the basic shapes that we're going to see, pay close attention to the arrangements of how these cells are put together. So the three basic shapes we're going to see this semester are cocci. So be very careful, that second C has a S sound. It's not cocky, it's cocci. That's plural. You also may see this semester. Coccus is singular. Bacilli, again, that's the plural form. You may see it like this. Bacillus is singular. That means rod shape, whereas cocci means rounded in shape. And the third basic shape is spiral. So now, folks, on an exam or in the lab, I would expect you to use these terms. I don't want you to just describe a cell as being round or a cell as being rod-shaped. I want you to actually use the terms cocci and bacilli. And we're going to see this is going to be very important as we start to identify bacteria by name. They're often named after their shape. So if you know what the terms cocci and bacilli mean, when I give you the name of a bacteria, you're right away going to tell me what the shape is. So for example, if I tell you, this is a bacteria you may have heard of before, MRSA. The third part here, the S stands for Staphylococcus. Right away, when I give you the name of the bacteria, Staphylococcus, you can tell me, hey, Professor Weber, that's a rounded bacteria. Without having to look at it under the microscope, you already know the shape. So it's a benefit to you now to learn the basic shape so we can apply it as we go forward. So let's take a look at cocci first, those spherical cells. Usually round. When we look at them in the microscope, they tend to be symmetrical. That means they're perfectly round. Now, some of them can be oval or a bit elongated, but for the most part, we're going to see perfect spheres. Now, interestingly, because of their shape, they can, when they divide, when one cell splits into two cells, they can split along many planes. Here's what I mean by that. If they divide along a single plane from top to bottom here, they often remain attached after they're done. So each cell, after it divides, represents an individual bacterial cell, but often they could be tethered together. So if a bacterial cell, in this case, divided one time, it would form a diplococci arrangement. Again, foundation slide. Learn the arrangements. Diplo means two. And if you look closely, it has Di, which often is associated with pair. So here I have a pair of cells. That's a diplo arrangement. Because the cells are rounded, that's a diplococci arrangement. Now if the cells continue to divide down this plane, they begin to elongate in either direction. If you look at it, it looks chained. We call that arrangement strepto. Strepto means chained. Streptococci means chains of rounded cells. Now, I want to pay special attention here. I want to point something out. When I write streptococci with a lowercase s, that describes shape and arrangement. However, this semester you're also going to see uppercase S, streptococci. It'll also be italicized, although it's hard for me to draw italicized on my whiteboard here. When you see uppercase S and you see it italicized, what that tells you is that's actually the name of a bacteria as well. Strep throat is caused by the bacteria streptococci. So streptococci can be a shape and arrangement, and in some cases can also describe the name of a bacteria. We often call that the genus name. Now here's what I would like you to do. I want you to go to chapter 10, and I want you to look up the genus and species name. in the part of the chapter which they discuss the hierarchy, the nomenclature, how we describe bacteria. I want you to go and look at what does a genus name, what does a species name mean. And I want you to be very careful this semester. Shape and arrangement, streptococci, lowercase s, is one way to describe bacteria. It's a general way to describe bacteria. But when I use the genus name streptococci, that's a very specific name for a bacterial species itself. All right? If you have questions on that, please ask me in lab. Now, what happens if it can divide along different planes? What if it can divide along two planes? Well, what happens? It can form sort of this layered arrangement called a tetrad. Tetra meaning four. We have 4 cells here. Now, I'll be honest with you, we won't see too many tetrads this semester. There are some species of Streptococcus that can form tetrads, but we often won't see that. The one I really want to point out here is the next one. What if you can divide lengthwise and widthwise? What if you can divide along 3 planes? What happens is you can grow outward. You can also grow in this direction as well. What we end up forming is sort of this clustered arrangement. That clustered arrangement is described by the prefix staphylo. Staphylo means clustered. So if someone describes an arrangement as staphylococci, what that means to you is you have a series of rounded cells, here we go, that form these clusters because they divide along many planes. Now here is another example. Staphylococci is a shape and arrangement, small s, big S, staphylococci. Or better yet, big S staphylococcus is actually a genus name of a bacteria. Staphylococcus aureus, for example. So again, please pay attention. Don't make the mistake this semester of thinking that Staphylococcus is only a genus name. It also is a shape and arrangement, or a way to describe shape and arrangement as well. Next would be the bacilli, the rod-shaped cells. You can see they look much different. They're much longer than they are wide. They look very much more elongated compared to those cocci-shaped cells. Now, many bacteria just exist as a single bacillus. What that means is after they divide, they don't stay attached. Examples we'll see this semester include things like E. coli and Pseudomonas. Those would be examples of bacteria that exist as single bacilli. We can also then, as they divide, they only divide along a single plane. So rod-shaped cells only divide along a single plane. So they can't be clustered. However, they can remain attached and form diplobacilli, diplo meaning 2. We can have streptobacilli. Remember we talked about this before. What does strepto mean? That's right, strepto means chained. For example, the causative agent of anthrax. The genus name for that bacteria is Bacillus anthracis. Bacillus, big B here, is the genus name. Anthracis, you notice lowercase, that's the species name. That's very specific. Notice they're italicized here. Bacillus anthracis not only is the name of a specific bacteria, it also describes the shape of the bacteria under a microscope. Actually, if you looked at this one, it would often look strepto in its arrangement, meaning these are large rods that stay chained together. The fourth type of bacillus, which creates problems for us when trying to identify under a microscope, is coccobacillus. Notice they have more rounded edges, but they're more elongated than just the cocci cells we saw previously. Can everyone see here's that perfect circle we talked about, where we can see these are rounded, but they're not perfect circles, right? Examples of coccobacillus would be, for example, Klebsiella pneumoniae, the name of the bacteria. We'll see that this semester as well. The last shape is the easiest of the 3 because it's more descriptive. Spiral shape, meaning it has more, one or more twists. Now there are 3 types of spiral shapes that we're going to see. Vibrio has a single twist. If you've been reading the newspaper in Haiti, they've had terrible cholera outbreaks. That's actually caused by a Vibrio-shaped bacterium. Spirilium has 2 twists. Here we go, one there, one there. We could see a nice actual picture of it. You can see the 2 twists. And then spirochete has many twists. It almost looks like a worm under a microscope. This is actually a picture of Treponema pallidum, which is the causative agent of syphilis. And interestingly, we're going to learn, I'm going to overemphasize the twists here, we often can describe it like a corkscrew. And what this bacterium does is as it invades tissue, it twists using an axial filament. You could actually see an axial filament right here. That axial filament actually aids to attach itself to that surface, and then it begins to rotate in this direction like a corkscrew, and it goes deeper and deeper into tissue, causing the infection that we see, for example, with syphilis, how it can spread, for example, to the brain. So those are the 3 shapes and and the arrangements that we're going to see this semester. Please take the time to learn them. It's only going to benefit you as we go forward. All right, so here's what we have. Here's an actual gram stain of urethral discharge. So we've taken a sample from the reproductive area. We've stained it under a microscope, and now we're looking at the shape and arrangement. I've actually tried to highlight them in blue circles here. Those are diplococci. Di meaning 2. You can see they're arranged in pairs. Some examples down here. And you can see their rounded shape, which makes them cocci. So again, this has real-world clinical application. I just diagnosed gonorrhea. And the reason I can tell you that's gonorrhea is partly due to the shape and the arrangement of those cells. I know the agent that causes gonorrhea is called Neisseria. Neisseria is a diplococci bacteria. So that's it for an introduction to cells and then an introduction to shape and arrangement. We'll talk more about other parts of the bacterial cell here as we go forward.